Welcome, everybody. It's a real pleasure to introduce George Tofaris, who I've known for over a decade. He's one of my colleagues at the Nuffield Department of Clinical Neurosciences, but he's also now working in the Kavli Institute. George is Professor of Neurology and Translational Neuroscience here at Oxford. He's also an MRC Senior Clinical Fellow and a consultant neurologist at the John Radcliffe. And he's been doing lots of interesting work on molecular aspects of Parkinson's disease. And today he's going to be talking to us about the prodromal phase of Parkinson's. Over to you, George.
Thank you, Masud. Thank you for the invitation and the opportunity to share our latest research. What I would like to do over the next 40 minutes or so is to give you a little bit of update on what we have done in terms of molecular understanding of the disease and how we're trying to translate that into a biomarker for the stratification or definition of Parkinson's and related diseases. Parkinson's, as you all know, is the second most common neurodegenerative disease. It's primarily characterized by movement disorder, the tremor, the stiffness, the unsteady gait, which is caused by the loss of dopamine neurons in the substantia nigra here, which is typically black, and that pigment is lost. This part of the brain, just to orient you, is on the top of the brain stem. We can measure this in live with a DAT scan, which essentially in patients demonstrates this deficit compared to a normal scan due to the loss of the nigral projections to the striatum. But at the moment, we don't have really any other means of identifying people who may have changes that suggest Parkinson's, and this test by itself is not diagnostic. Of course, we know that Parkinson's is much more than a movement disorder. It's characterized by what we call non-motor symptoms, in particular loss of sense of smell, changes in mood, apathy, autonomic problems such as constipation, bladder trouble, and a sleep disturbance, something called a REM sleep behavior disorder. And we know now that a number of these non-motor symptoms start in this phase here, what we call the prodromal phase of the disease. That is a phase during which the pathological cascade is sort of initiated and sort of leads to progressive pathological change in the brain, but the patients are sort of coping and have not yet manifested with a classical movement disorder to the clinic, which let's say is here. So the question really is also, when does this process start? We have no idea, and I'm not going to claim that I'm going to solve this in this talk, but I just want to raise some sort of interesting points about how we are approaching this. So essentially what I'm hoping to do is to discuss some key pathogenic processes that I think are relevant to the prodromal phase as well as the clinical phase of the disease, and also discuss how we can use this fundamental knowledge to progress towards a molecular definition of the disease and a stratification of the patients. Now, Parkinson's and related diseases are complex and multifactorial, but a key event in this disease is the accumulation of alpha-synuclein, a small protein that is found normally in the synapses, but forms these abnormal aggregates in the pigmented neurons I mentioned earlier in the substantia nigra. And these aggregates, called Lewy bodies, are made of these filaments, which we now know have sort of definition at a cryo-electron microscopic level, are made of alpha-synuclein aggregates. And this process is thought to arise from the conversion of soluble synuclein, let's say these a blue dot here, into a misfolded conformer that has a beta sheet sort of structure, and that in turn acts as a template to convert more soluble synuclein into the insoluble form, and these misfolded conformers assemble to form fibrils similar to the ones you see here, and then they sort of clamp up to form the aggregates you see here, and trap a sort of a number of other sort of organelles and disrupt the cell. And this process is thought to evolve, and according to some literature, spread along interconnected networks of the brain that seem to correlate with a progressive march of clinical symptoms in the patients, suggesting causality. These have not been retrospectively shown, but it's suggested also from animal models. Another key sort of evidence from humans is the presence of mutations in alpha-synuclein gene, and most importantly, duplications and triplications in the alpha-synuclein gene, which suggests that a slight increase in the levels of alpha-synuclein, and we're talking here about a twofold increase, is associated with increased severity of disease, high risk of conversion to dementia, and an earlier age of onset. So again, suggesting a causative association between alpha-synuclein and Parkinson's disease. So, and this sort of initial work that started, I guess, with the discovery of synuclein, the discovery of synuclein Lewy bodies, the genetic mutations, led to the first phase, if you like, of the molecular classification of these diseases into what we call now alpha-synucleinopathies, which beyond Parkinson's also include Parkinson's dementia, or also dementia with Lewy bodies, which is a condition characterized by the same aggregates primarily in the cortex, and presents initially perhaps or predominantly with dementia. And then on the other end, a condition we call multiple system atrophy, which is characterized by alpha-synuclein aggregates in the oligodendroglial cells that surround the neurons. And both of these conditions are associated with neuronal degeneration and loss of the dopamine cells in the substantia nigra. But the kind of type of assembly of alpha-synuclein into these filaments has slightly different conformation, suggesting that the conformation is quite critical in how toxicity is determined. Now, there is a lot of evidence for that kind of statement I just made, but I just wanted to divert slightly and point towards a recent study we published to make that point. And this is where we essentially take advantage of this process that we think is occurring in cells to induce aggregates derived from iPSC cells, in human neurons derived from iPSC cells, and sort of determine the significance of this event in a human neuron. Others have done this in animal models, but we were the first to do this in human dopaminergic neurons. The way we did this is to take iPSCs from either health controls or patients with these triplication of alpha-synuclein gene that I mentioned earlier, and add the exogenous fibrils. And what that does is induces aggregation inside the cells, which we can monitor with a pathological synuclein staining pattern, stain using phosphorylation at 129. And you can see that the pattern of pathology here resembles what we see in the human postmortem brain, with neuritic processes as well as cellular aggregates. And what is important here is that the presence of a duplication or triplication in this case, which is only a twofold increase in synuclein levels, leads to an exponential increase in the number of aggregates with time and culture compared to the healthy controls here. So what this says essentially is that how much synuclein there is in the cell, and the difference is, as I said, about twofold, is key in determining the extent of aggregation. That's perhaps not surprising. There is evidence from animal models to show that the pattern of spread of pathology sort of follows regions that are highly expressing alpha-synuclein. So one event is the amount of synuclein. The second key determinant, I guess, primarily for toxicity, is the aggregate structure. And we sort of were able to demonstrate this by taking advantage of the conformational plasticity of alpha-synuclein by generating essentially fibrils that are more or less toxic, and also not any fibrils, but fibrils extracted or amplified rather from brain homogenate, either from Parkinson's disease or MSA, which is a more aggressive disease, and add those in a fairly pure form onto this human iPSC-derived neuronal system that I have described, and monitor sort of the toxicity and aggregation. And what we found in short is that essentially MSA, which is a more aggressive disease, leads to slightly different types of aggregates in neurons and culture compared to PD, which are different from recombinant fibrils generated in vitro. But the important point here is that MSA, which is more aggressive disease also leads to a more extensive cell death in the dish compared to PD, as measured here by a lot of dopamine cells, and here by the release of neurofilaments. And each dot here is a clone differentiated once with multiple sort of images. So it's a quite a robust readout. So the point here is also that by reducing synuclein back to normal levels, in this case by using what we call an isogenic control, the same clone of cells that have been CRISPR engineered so that the levels of synuclein go back to normal, not removed completely, we can essentially reverse the extent of aggregation, and that reverses the toxicity. So essentially what I'm trying to say is whether a cell dies depends on two things primarily: how much synuclein there is in that neuron and what conformation it acquires when it aggregates. And different conformers, as we showed in this paper, for example, can differentially cause toxicity by, let's say, evading protective responses, or perhaps being more strongly interacting with certain detrimental, so certain functions of the cell which are detrimental. So if this is really key, and this is supported also by animal studies, clearly understanding how synuclein is handled by the cell, especially when it starts to aggregate or even beforehand, might be quite important in eventually developing therapies, which is not the purpose of this talk, but also developing biomarkers. And in our lab, we're guided to these by sort of these three sort of axes here. We have already discussed the importance of alpha-synuclein and its aggregation. Another important clue is the presence of ubiquitin around these sort of protein aggregates in the dopaminergic neurons and other brain cells, and the genetics which implicate lysosomes in particular, and this endosomal lysosomal pathway in the disease process. So how can we bring all these things together? And throughout the years, my research and our group's research suggested that ubiquitin signaling might be a link between alpha-synuclein homeostasis and these endosomes and lysosomes that are implicated in disease, and might be the accumulation of ubiquitin when this process fails in Parkinson's. But I wanted to briefly mention essentially that we have now initially sort of did this by biochemical approaches using specific and manipulating specific enzymes, but more recently, in a paper that is in press, we were able to visualize the ubiquination of synuclein in living cells and monitor its fate in a fairly unbiased fashion. And this is taking advantage of what we call a complementation assay, essentially an assay which makes synuclein fluorescent when it's conjugated to ubiquitin, and by mutating that sort of ubiquitin or the synuclein, we can sort of have control conditions to ensure that this is specific. And here you see in live cells the ubiquitinated fraction of synuclein. This is the conjugation of synuclein to ubiquitin chain, which essentially is highly reversible and dynamic. And this is quite reassuring for us and exciting, because we can now follow its fate. And I'm not going to go through the paper in detail, but through proteomics and live imaging and further colocalization studies, we're able to show that once synuclein is ubiquitinated, it ends up in these sort of red dots here, which you can see here, the fusion of the green dots with the red dots. That means that the ubiquitinated synuclein goes into the endolysosomes for degradation. Here is further images in fig cells where you see synuclein in these dots colocalizing with components of the endolysosomal system, but not with mitochondria or the Golgi ER sort of trafficking steps. So what I'm trying to say here is that when synuclein is non-aggregated, it goes through a ubiquination into endosomes for degradation by lysosomes. This is just a kind of a further evidence to demonstrate that these signaling that we have delineated is very specific and selective. What we did here is we did mass spectrometry to identify the specific lysines on alpha-synuclein that become ubiquitinated and we found only 3, despite the fact that synuclein has 15 licenses. And when we mutated those 3 licenses, we completely lost the signal. So essentially, the signal I showed you before, those green dots, is highly specific due to the ubiquination of synuclein on this specific license. And it's also abrogated when we mutate the ubiquitin in a way that can no longer be conjugated to alpha-synuclein. And then again, here you see that when we mutate these 3 sites, we lost this smear, because this is what happens when protein is ubiquinated and we lose these dots inside the cells. And I want to just point out this corroborating assay, where essentially we express alpha-synuclein that is conjugated now to a pH-sensitive GFP, which enable us to visualize synuclein that enters acidic compartments such as endosomes and lysosomes. And we have here the wild-type synuclein, or the synuclein mutated to these 3 key lysins that I mentioned here. And what we have observed is indeed we can visualize here after addition of chloroquine, which essentially neutralizes the acidic pH in this dilated endosome, which we dilate by expressing a mutant RAB, and that enables us to see what's going on inside, and that is sort of lost when we have the mutation. And all this data essentially suggests a highly specific way by which synuclein is localized to endosomes after ubiquitination. And I just wanted to point out that this is not just an in vitro assay, but it's something we demonstrated to happen endogenously in cells. And we did that by generating an antibody, and we are now working with the Michael J. Fox Foundation to make this available to the wider community. Essentially, this antibody recognizes these 3 ubiquitinated lysins, and as you can see here in an in vitro reaction, it recognizes that smear, but not the monomer of synuclein. And this antibody recognizes dots in cells. This is endogenous now, neuronal-like cell lines. And these dots disappear completely when we engineered the cells so that they no longer express synuclein. That's very different from what we're used to with synuclein antibodies. This is the commercial antibodies that see the diffuse staining, whereas with these ubiquinated antibody-specific, ubiquination-specific antibody, we see only the dots. And here is again the biochemistry here to see that there is a smear that disappears. And that is seen also endogenously in primary neurons, because we have seen again here these dots that colocalize with endolysosomes, the LAB1 positive structures, and that sort of signal disappears when we add MLN7243, which is essentially an inhibitor of the ubiquination reaction. So we can block that from happening. The smear disappears in a lysate, and the signal of those dots disappear in cell culture, demonstrating essentially that the mechanism that I have been describing is occurring also endogenously in neurons. And this is the first demonstration of such an event. Now, why I have been going on about this for such a long time, I wanted to explain how we eventually got to the patients by the in-depth understanding of the molecular trafficking of synuclein and how it's handled by cells. So essentially, in a diagram here, what I showed you some of the data we have to indicate that synuclein gets ubiquinated, is conjugated to these ubiquitin molecules. That triggers is localization to endosomes. I didn't describe all the key cofactors here, but that what that does essentially, it internalizes synuclein into endosomes, which is then fused with lysosome, which is a kind of a bag of digestive enzymes to chew up synuclein and normally in cells. And because in Parkinson's, as I mentioned earlier, there are there's evidence from genetics that lysosomes are not working very well, we reasoned that under perhaps conditions of impaired lysosomal function, alpha-synuclein can be released outside from these endosomes into these extracellular vesicles, which are endosome-derived structures. And we know that if we block lysosomes, for example, there is increased release of exosomes in cells. But what we wanted to do is to see whether we can show that in patients. And most importantly, not to study any extracellular vesicles, but extracellular vesicles derived from neuronal tissues, or ideally the brain. And we can do that in principle because we now know that there are different types of these nanoparticles in the circulation that are derived from different types of cells or tissues, and we can track them by identifying those that express on their surface markers that sort of correlate with the site of origin. In this case, will be L1 CAM on the surface of neuronally derived extracellular vesicles. So what we want to do is to develop an immunocapture method to isolate from the circulation those EVs or extracellular vesicles that are neuronally derived, and develop a proxy biomarker that could potentially tell us whether this is true and whether this fails, and when this might fail in disease in using patient samples. I just wanted to show brief evidence that indeed, first of all, synuclein is found in association with extracellular vesicles in the circulation. And we did that by using serum that is passed through a column, which enable us to fractionate the serum in 10 fractions and separate essentially the extracellular vesicles, which are measured here with something called nanotracking analysis. It's a way to visualize small particles. You can see them in region fraction 3 and 4 from the bulk of free serum proteins that are found in later fractions here. And this is a Western blot to show that indeed in these two fractions where we see vesicles by imaging, we also have enrichment of these two extracellular vesicle protein markers. But what is interesting, though, from this data is that when we look at alpha-synuclein, the majority of alpha-synuclein is free in the later fractions, whereas a tiny portion, which is about probably less than 50 picograms in a ml of serum, is found in association with these extracellular vesicles. And bear in mind, these are all the vesicles, including neuronal and non-neuronal. And here in blue, we show another marker, CD81, which is another marker of extracellular vesicles. So essentially, this fraction here, which correlates to these two F3 and F4, has a small amount of alpha-synuclein, which is quite tight, actually. It's quite tight, the amount that you detect here, despite actually variation in the free amount of synuclein. And this is because alpha-synuclein is primarily in serum in a free form derived from non-neuronal cells, primarily red blood cells, which are prone to hemolysis. So you can have tons of synuclein essentially in the serum, and a tiny fraction would be associated with extracellular vesicles, and an even smaller fraction of those with neuronally derived vesicles. So to be able to capture those specific ones in the unfractionated serum, which is what we wanted to do to study thousands of samples or hundreds of samples, we develop an improved immunocapture assay, which essentially involves the coating of magnetic beads with a polymer that which is then linked to an antibody. In these cases, the antibody is against L1 CAM, this neuronal antigen that I mentioned earlier, to capture L1 positive extracellular vesicles with minimal fouling from other free proteins or free alpha-synuclein. And this is just to show that indeed, when we synthesize the beads, we could show that our beads compared to commercial beads are less sticky. Here is incubation with serum and then a silver stain to show that there is much less contamination compared to what you get with a commercial beads. And here is also the evidence that if we incubate this sort of a beads conjugate to an antibody with recombinant alpha-synuclein, we get very little binding of free synuclein, whereas with commercial beads, we get a higher amount. So I think this is quite a key to what I'm going to show from now on, and very different from some other attempts by other labs, which may have shown slightly different results. So this is just to show that in those fractions that I mentioned earlier, fraction 3 and 4, we can immunocapture neuronally derived vesicles using L1 CAM. And you can see them here by electron microscopy. These are cap-shaped vesicles that are not detected in controlled conditions. And when we quantify them, I can tell you that they're about 10% or less, between 5 and 10% of all those vesicles that are found in fraction 3 and 4 in the serum. So, and in healthy individuals, here you can see the average amount of synuclein in healthy people in their neuronal-derived vesicles is about 10 to 15 picograms per ml. As I mentioned earlier, there are about 50 picograms in total in all the EVs in that fraction, and about 10 or so are associated with neuronal vesicles, which are less than probably 10% of all vesicles. So there is some enrichment probably of synuclein, which are, after all, also a neuronal protein. And here it's just to briefly mention that essentially we went on to do, let's say, a few controls, and we can see that our assay now is quite consistent. Top we're showing synuclein, the bottom is synten, which is a generic exosomal protein, to show that indeed we can capture these vesicles and measure synuclein consistently when we test two aliquots separately, when we extract them 6 months apart, or when we sample the same person, health individuals, 18 months apart. So that was quite encouraging for us, suggested that we do have an assay that we can use reliably to interrogate patient samples. So the first thing we wanted to do, having essentially established the assay that we think reflects some of the biology we're investigating in earlier years and more recently with the study I have just shown, was to first study Parkinson's patients versus controls using 3 different cohorts from Oxford here, the discovery cohort from Italy and Germany. And what you can see here using the assay I just mentioned is that alpha-synuclein in neuronally derived vesicles from Parkinson's patients is higher than the controls. Each dot here is an individual tested once. And when we define a threshold based on this separation and we apply it to the validation cohort, in this case, I think it was Italy and Oxford, and then German cohort here, and then Oxford and Italy here, you can see the very consistent performance of the test with an area under the curve of 0.86, which roughly means that 86% probability of separating those who are positive from those who are negative based on this cutoff here, which is 14.2 picograms. This is essentially roughly the same accuracy as that of a neurologist or a general neurologist, at least, diagnosing Parkinson's disease. So what we were able to do next, which is I think more important for clinical application, is to show that our assay can very nicely distinguish Parkinson's from Parkinson's-like conditions. And this is now involving this number of patients from 4 cohorts. Some of them were the same as before. But what we're able to show is essentially what is quite interesting. PD is very separate, different from MSA in this regard, because as I mentioned before, in Parkinson's disease, we have neuronal synuclein dyshomeostasis and aggregation, whereas in MSA, we have primarily glial synuclein accumulation. And what we think we are doing is measuring neuronally derived vesicles in serum. And it's quite exciting that in MSA, which is not a primarily neuronal disease, we do not see the same results. So that's why we're able to separate PD from MSA with a very high area under the curve between, again, a training and validation cohorts. This is a cross-group validation with an area under the curve of more than 0.9, which is quite remarkable. We are, of course, able to demonstrate that we can separate the synucleinopathy, the neuronal synucleinopathy, from other proteinopathies such as 4-repeat tau, and these are conditions like PSP and corticobasal syndrome, which can mimic Parkinson's disease sometimes. So I think it's quite useful to have an assay that perhaps can confirm the clinical suspicion and exclude the more aggressive forms of Parkinsonian syndromes, perhaps in the future. Especially, this is important when we come to stratify patients for disease-modifying therapies that target neuronal alpha-synuclein homeostasis. But what I'm most excited about, which is also coming towards perhaps the core of my title, my message on my title, is the prodromal phase. What happens in the prodromal phase? What can we detect? And what does it mean? So we have done recently a prodromal study, and that was done by Cheng and Victor in our group, involving more than 350 people at risk of developing Parkinson's disease, again, using the Oxford Discovery group here that is enriched particularly in people with REM sleep behavior disorder, which, as I mentioned, is one of the highest risk for Parkinson's and related diseases RBD, as I mentioned earlier, increases the risk of Parkinson's by 6.3% per year, and this is cumulative, which means that people with RBD for 15 years will have more than 80% chance of probability of getting Parkinson's. So we have RBD from Oxford and independent groups from Marburg and Cologne, and then we have a separate at-risk cohort from the PPMI. This is the Michael J. Fox funded multi-center cohort, which involves different individuals with variable risk, such as people with RBD, but also people with hyposmia and an abnormal DAT scan, people with GBA mutations, etc. And we have some controls as well, some Parkinson's controls. So what we wanted to do is to define cutoffs again and cross-validate across these groups. And I think it's quite exciting to show that, as we observed in a slide earlier, a sort of pilot study, that indeed we can separate the RBDs, these are the people at risk of developing Parkinson's, from controls by measuring alpha-synuclein in their neuronally derived extracellular vesicles. And when we define a cutoff here, a threshold, which is applied to the German groups, and this is shown by the blue line here, we can still see a very nice separation with an area under the curve between 0.84 to 0.9, which is quite remarkable. But what is quite exciting is that if we apply then the same threshold from here to the PPMI group, which I mentioned, these are individuals with variable risk, we can still separate those who have a high probability of more than 80% of developing, being in the prodromal phase of PD, from those who have a very low risk of less than 5%, which is essentially close to normal risk, using our blood test. And this is, just to clarify, this sort of probability here is based on a number of research criteria, which are themselves based on a number of detailed, labor-intensive clinical assessments. So with a single blood test, we can achieve almost the same result. And here are the Parkinson's from this group to confirm that indeed the assay is working. As goes without saying, all of this was done in a blinded fashion, and then blinded by the Michael J. Fox. So it's quite exciting, rewarding to see this separation. And here, what we have done is to just put all the patients together and separate them based on their sort of clinical assessment and probability scores into those who have a positive likelihood ratio of having prodromal Parkinson's, which is defined as more than 80%, from those who have less than 80%. And here are a number of healthy controls we analyze from this study and throughout the years. And I think what you can see in this overall assessment is that we can identify, again, those with a high probability of having prodromal PD with an area under the curve of 0.9, which is quite a first, I think, for a blood test in Parkinson's. And here is the generic protein syntenin-1, which is not changing across these populations as we would expect. So having shown that synuclein is released in these extracellular vesicles, it seems quite early in this cascade that leads to eventual pathology, we wanted to try and correlate with some other markers or biomarkers and see whether we can figure out when that change might happen. And of course, it's different in life to do this for neurodegeneration pathology, but there are ways around it using imaging and other assays. So I'm going to just share some of these results on the next slide. So first, we asked how does synuclein in L1EVs, these are the neuronally derived extracellular vesicles, changes in people who are at risk, not with Parkinson's yet, but have a positive DAT, an abnormal DAT scan or a normal DAT scan. Individuals with an abnormal DAT scan are more likely to go on to develop Parkinson's than those with a normal DAT scan that I mentioned earlier. And here you can see that indeed those with an abnormal DAT scan had higher synuclein overall and separated from the healthy controls with a higher area under the curve as expected. But what is interesting here is that a number of individuals who are at risk with a normal DAT scan also had high synuclein levels in their serum L1EVs, suggesting perhaps that these before the neurodegeneration is seen, or at least as measured in live by a DAT scan. So that is interesting. And we also investigated the relevance of our blood test in relation to pathology as measured by what we call a synuclein seed amplification assay. This is an assay of spinal fluid in the CSF, where essentially uses some of these prion-like sort of templated mechanism that I mentioned earlier to measures the presence of minute amount of aggregates, because CSF with minute amounts of aggregates incubated with recombinant synuclein will lead to formation of large amounts of aggregates that can be measured in this assay, essentially. And you can see that in the PPMI cohort, those who have evidence in their spinal fluid of the aggregates had a higher amount of L1 EVs detected in their serum compared to those with a negative amplification assay. And of course, when we also look at this in relation to the probability of developing Parkinson's, you can see down here in less than 5%, there was a good correlation, I would say, between those that are negative for the CSF assay and the L1 EVs in the serum. And on the other end here, the vast majority, about 76% or so of those who had a positive assay also had a positive alpha-synuclein in their serum, which is quite good. You might say, why is this sort of variability in the middle here? Well, I would like to point out that these people are already at risk of developing Parkinson's, and we do not know at this point which one comes first. Is it L1 goes first and then you become positive for pathology, or vice versa? We also don't know whether SAA negative patients do not really have pathology, because according to the latest study, only about 86% of those at risk had a positive test. So it's not like SAA is as 100% assay. But I think, again, it's possible that although the majority are negative and below the threshold, this is, by the way, the line here is the cutoff that we have defined earlier on, a number of people may be developing changes in that are detectable in blood before the detection of pathology as measured, at least by this assay. And what is actually quite exciting is the ultimate validation of the test. Obviously, this will require a further longitudinal assay studies and assaying in people who went on to develop the disease. But what we were able to do is to go back to the data and identified out of the 350 at-risk individuals, 40 that converted to disease. And you can see, based on the threshold here, that about 80% or so of those who went on to develop either Parkinson's or dementia with Lewy bodies, which are both neuronal alpha-synuclein pathologies, were correctly identified and positively identified by our blood test 1, 2, or 7 years before they developed this got this diagnosis. And one individual in this cohort, which of course is a PDN cohort, went on to develop Alzheimer's, and that individual was below the cutoff, which is quite reassuring. So if we look at the data for those that converted to Parkinson's, you can see a weak, but nevertheless a weak positive correlation between the amount of synuclein that is released in the extracellular vesicles and the time it takes to convert to disease. What this means, essentially, in other words, is that the more synuclein is released outside in the circulation, the longer it takes to convert to Parkinson's disease, essentially suggesting that this may be a protective mechanism, which would make sense based on the biology that I described earlier, that is, synuclein is trafficked through to the lysosome, and when that process fails, and since there is a way out, maybe the cell is taking advantage of that mechanism to release synuclein outside. And just a little bit more here to show further support for this statement I just made is the detection of a weak but inverse correlation between the amount of L1 alpha-synuclein in the serum and the total amount of alpha-synuclein in the CSF. This is not about aggregates here. This is just total CSF synuclein. And what we can see here is that there's a tendency, the more there is outside, the less there is in the CSF in those who went on to convert to Parkinson's and people with sporadic Parkinson's disease. So, and this would fit quite nicely, I think, with some of our earlier observations in our first paper in the clinical phase of Parkinson's, where we observed a consistently elevated levels of synuclein in those with Parkinson's compared to controls as they progress, and which sort of mirror imaged what Mollenhauer and colleagues reported in the CSF of Parkinson's patients and at-risk individuals compared to controls. That is, there is an increase in the serum synuclein in these neuronal vesicles and a reduction in the total CSF synuclein. And that remains the case with the progression of the disease. And I think what we just showed you with specific cases is there is some correlation in a one-to-one basis. So essentially, as I mentioned, if you just imagine again, here is the brain, this is a neuron and neuronal tissues, let's say here is the CSF, and here is the blood. What we are really measuring in these nanoparticles, these that are generated within the endosomes, what we call multivesicular bodies, they go out, they have a surface neuronal marker that we can identify and capture. And I think this means essentially is that the L1 positive particles offer a means of reading the cells of excess alpha-synuclein under pathological conditions that we can actually measure in the circulation. So although there is a lot of interest in the definition of Parkinson's by biology, a lot of the emphasis, I believe, currently is on these amplification assays in the spinal fluid measuring aggregates. And I think there is a kind of a problem with this because, first of all, we don't know when this starts. It's very hard to know when aggregate starts because when that is limited by the detection of the assay, limit of the assay, but also the window, I believe, of detection to degeneration or clinical symptoms is quite narrow when it comes to thinking about aggregates. And we need to extend the solvable sort of prodromal phase. And although I haven't quite demonstrated that experimentally, I believe that our data suggest that there is some sort of a fundamental shift in synuclein homeostasis that is probably reflected to that switch from the CSF going down and the L1 going up. And that is probably, I would say, the beginning of, or at least the measurable beginning of a change in synuclein processing that is going to lead eventually to the detection of aggregates and neurodegeneration. And I believe that that is probably a phase we need to define more accurately and identify, because that's the solvable phase and more like where precision therapies against alpha-synuclein might be more effective in the longer term. So I just want to finish off by just mentioning that we have also spent a lot of time and effort with our colleagues in chemistry led by Jason Davis there to not only develop the biomarker, but also develop the tools and the technology to make this scalable and suitable for an eventual screening test or a test that can be used across laboratories reproducibly to measure these changes. So what we have recently managed to do is to develop an on-chip capture of extracellular vesicles by sort of injecting into these microfluidic chamber a serum sample with beads conjugated to L1 antibodies. And the trick here was to apply magnetic agitation, because this enables mixing and capturing of vesicles whilst reducing non-specific binding, and then using a permanent magnet, immobilize the beads and the vesicles on them, wash through the flow through, and then measure electrochemically, which is a kind of a different way from what we have done before, the content of those vesicles. And what we can do now is to do essentially what we can do in the lab over a period of 24 hours or so, within 30 minutes, at least in terms of capture, from 50 microliters of serum, with a capture efficiency of more than 80%. And when we do this, I haven't included this data, but essentially we found the same result, that is synuclein is increased in PD and RBD. So essentially, we want to move towards an eventual test that can be used to identify, ideally, I believe, people at risk of developing Parkinson's in the same way we now perhaps do for certain cancers, like prostate cancer with a PSA or breast cancer with an X-ray screening. And this is going to be quite important when we have, and we will have one day, disease-modifying therapies targeting alpha-synuclein that slow down the disease. It's also quite useful to have a test like this when stratifying patients that have primarily a neuronal synuclein pathology, because we know from failures in Alzheimer's field that unless patients are clearly well selected and stratified, we might compound the final outcome of any tested therapy. So I would like to end there. Thank the group. I would like to especially acknowledge Victor and Cheng for the biomarker studies, and Dimitri and Jade for the, and Andrew here, for the cellular work I presented. All my collaborators, funders, and thank you for your attention. I'm happy to take any questions.
Great, thank you very much, George. Fantastic. We've already got some questions in the chat, and I'm just going to encourage people who are there to put some more questions in that we can ask you. So George, Nick Wilcox has a couple of questions. He says, does the donor age make a difference? And are you working on an analogous assay for MSA?
You mean about, I guess, the donor for the serum assay? Yeah, that's a good question. Obviously, Parkinson's is primarily an age-related disease, so most of the patients will be around the age of 60 and above. We haven't really done a detailed analysis of the content of synuclein in L1 EVs with time, and certainly we should look at that. In terms of MSA, perhaps I went through it quickly. We have tested MSA, and L1 alpha-synuclein in MSA is not elevated, which makes sense in a way, because for two reasons, I guess. One, as I mentioned, because MSA is primarily a glial disease rather than a neuronal sort of pathology. And secondly, in Parkinson's, you have a lot of peripheral neuronal involvement, whereas in MSA is primarily a central sort of nervous system disease. So it's possible some of those L1 EVs that we detect in serum are coming from peripheral autonomic nerves. And I think these are the two reasons why it makes sense that we don't see the same in MSA.
Nick's followed up just asking, does the donor age of controls matter too? Right.
No. As far as we know, because we tested some of the our cells in the lab, the healthy volunteers I mentioned earlier, and those are quite similar to the controls that we have from cohorts, which are usually partners or spouses of patients. So we haven't really seen any difference, although perhaps we need to do a big study to be entirely sure about this. I think what's more important is how carefully the sample is prepared. We know that freezing, thawing the samples many times is not good, and in terms of hemolysis, I guess, is quite key.
And James V is asking, to follow on from that, would you expect similar stratification versus ethnicity and other comorbidities? I mean, do you
Yeah, that's an important point, I guess, that a number of these biomarker studies suffer from. That is, most of the work we do is involves cohorts that are generated in Western Europe or the US. Therefore, we don't have enough data really to look across ethnic backgrounds, and that's a limitation of ours and other studies in this area.
What about renal function? Does that make any difference?
The what, sorry?
Renal function, kidney function?
Yes, I don't know. That's another good point, because exosomes are also released in the urine and are detected there. We haven't really actually tested ourselves whether these L1 EVs are present in the urine, but there are lots of exosomes in the urine. And so maybe that's the eventual route of getting rid of these sort of particles and their content. And of course, if there's impaired renal function, that might affect the levels, but it's something probably we need to check.
Okay, and I was just wondering, where do you see, if you did have this test available clinically, where do you see it sitting? Where is it going to be useful?
Well, I think it's going to be useful eventually when we have disease-modifying therapies in identifying those individuals at risk. It could be a screening step to find those who are at risk. I was yesterday at the other day at the open day, and when we asked our participants, would you like to take a test if it could tell you whether you're going to get Parkinson's eventually, especially if there is a disease-modifying therapy, the answer is uniformly yes. So I think there is a need for that eventually. I appreciate now we don't have the therapies and it might be difficult. This test could be also used to stratify patients for clinical trials, I think, because as I said, it identifies those with neuronal synuclein pathology versus perhaps MSA or other diseases, and increased confidence, and might be used to reduce also the number of clinical assessments that are done currently to identify those at high risk. For example, when we did some sort of modeling, we can see that if we have a cognitive assessment with a MOCA, a sudden value of alpha-synuclein and RBD, then you can fairly confidently identify those on high risk of converting to Parkinson's. So maybe you can integrate a blood test that can be done very quickly with a limited number of clinical assessments to stratify people better, either for clinical trials initially and eventually for precision therapy.
Right.
I think those therapies will come. Obviously, they're not a bit, but they will.
Yeah. Another question here from Nick are you developing an oligoexosome assay?
Oh yes. So I assume you mean oligomeric synuclein or aggregates.
I presume
that is the question. Then we don't know whether there are aggregates.
No, I mean oligodendrocyte assay.
Sorry. Oh yes, the other people have done this, and there is some evidence that there are oligodendrocyte-derived vesicles in the circulation, and some of those might be useful in separating further MSA from PD. I think we haven't done this, so we're not working on this ourselves at the moment.
Okay. Any other questions? You're all being a bit shy. This is the time to ask. Anything else anyone wants to ask? Okay. And George, what about this distinction between DLB and PD or PDD in terms of the marker? Do you think it makes any difference which of these clinical phenotypes people classify them in terms of what you get from the assay?
Well, I think based on our results, and I showed you the conversion rates, we can not really distinguish at the moment between DLB and PD. However, in our previously published study and those who converted to PD, we observed that DLB cases had slightly lower synuclein levels compared to the PD ones. They were both higher than controls, but DLB was slightly lower. So whether it is possible to refine the test to separate the two conditions, I don't know. But at the moment, I don't think that we have enough data to say that we can do that.
I guess that brings up the question about where in the brain these exosomes are coming from. Do you think they're coming from everywhere, or do you think there's any sort of partial selectivity about where these exosomes are coming from?
Yeah, it's difficult to know. I think that's an important question. They may, as I said, they may be even coming from outside the brain, from nerve cells that are affected, like autonomic nerves, etc. So we haven't really shown that. The prediction would be, based on what I said, is from those cells that are more vulnerable and are accumulating synuclein. But I think we need to somehow study this experimentally in an animal model, for example. I don't know.
Yeah, no, I can imagine that would be really hard to work out. But yeah, fascinating. All right, thank you very much, George. Fantastic talk. and thanks to all our attendees as well. Thanks very much.